Till now we have studied about the asexual reproduction process in animals and plants. However, in flowering plants, the asexual mode of reproduction occurs, but this term is generally mentioned as vegetative propagation, which is generally used in common. Now, what is exactly the vegetative propagation? Vegetative propagation is a method of asexual reproduction, or rather it is a type of asexual reproduction in plants, mainly in flowering plants, where some part of the organ of a plant this plant is termed as mother plant, from which the organ is taken. For the development of a new plant. Now, the process or the method of vegetative propagation are of mainly two types. First, natural vegetative propagation, and second, artificial vegetative propagation. Definitely the name suggests that in natural vegetative propagation, the development of a new plant would take place under normal suitable environmental condition. On the contrary, in artificial vegetative propagation, a few environmental conditions would be created in order to obtain new plant. So let us understand both the types in brief with the help of examples. Let us first begin with the first type, that is natural vegetative propagation. This vegetative propagation can be also said as vegetative reproduction. As said earlier, in this development of a new plant occurs from any organ of the mother plant under suitable environmental conditions. This vegetative propagation, that is natural vegetative propagation, can be mainly carried out with the main organs of the plant. That is, as we all know that the main organs of the plant are root, stem and leaf. So it can be carried out mainly with the help of root, stem and leaf. This figure represents the example of the vegetative propagation by root. This is the roots of dahlia and this is sweet potato. That is, these are the plants which develop with the help of adventitious root system. This is the vegetative propagation by stem. As already studied earlier in 11th, tuber formation occurs in potato by vegetative propagation method. Here the tuber development occurs due to the formation of eye-like structures which are axillary buds of the stem which helps in the development of a new plant. And this is the example of natural vegetative propagation by leaf, where the development of buds from the leaf occur in order to produce new plant, for example bryophyllum and agave. This was all about natural vegetative propagation. Now next we will study about the artificial vegetative propagation. Now we will start with the study of artificial methods of vegetative propagation. Now these methods mainly include of two types. First, in fields, that is in farming field, they can be of three types or rather three methods. First, cutting. Second, layering. And third, grafting. This is in farming field. However, with the help of latest techniques, in laboratory also, vegetative propagation can be induced. And the method that is induced in the laboratory is called tissue culture. But here our main focus will be on these three methods that are used in the farming field. So let us begin the first method, that is cutting. In this method, the cut pieces of root of a plant, which here will be termed as a parent plant. So cut pieces of root of the parent plant are inserted into another plant that is placed in the moist soil which gradually with time helps in the development of adventitious buds. Thus helps in the root system development of the plant into which this cut portion is inserted. In this way, the root system of another plant will be developed artificially. Thus, as it can be seen from the figure, this is the parent plant from which the root is cutted with the help of blade. And after this process, that system is inserted into another plant which is in the presence of moist soil, which helps in the root system development of the other plant. And thus in this way vegetative propagation in an artificial method is induced. Example of plants which can be induced with the help of such methods is rose, sugarcane, chrysanthemum plants. Thus, in these plants, proper sizes of the stem are cut or obtained in such a manner that they can be inserted into another plant for root system development. Thus, from this cutted stem pieces, new adventitious roots develop. Thus, it helps in the development of a new. The so-called plant that develops in this way is called cutting. However, gradually with time, these cuts are transplanted in proper places. Now, the second method that we will study after cutting of artificial vegetative propagation comes layering. This method is generally employed in the cultivation of plants like rose, lemon, jasmine, grapes, and hibiscus. Thus, these plants develop with the help of layering method. But how does this process is executed? This figure depicts that how a plant can be cultivated or obtained with the help of layering method. As it can be seen from the figure, the lower branches of the plant are bent in such a manner that they are pressed under the soil, in such a way that the tip of the branch remains above the soil and the middle portion remains down, that is below the soil. When such adventitious roots develop from this buried region, then the branch is cut and separated from the parent plant. Thus, a new plant is obtained with the help of this method. Therefore, in this method, lower branches of the plant are bent in such a way that the tip of the branch, that is this portion, remains above the soil. And the middle portion, that is this one, remains below the soil or below the ground. Thus these branches remain buried under the soil. Gradually with time, adventitious roots or adventitious buds develop from this middle portion or which is buried under the soil. So, when adventitious roots develop, from this buried region, then this branch portion is cut, separated and inserted into another parent plant. Then the branch is cut and separated from the parent plant. Thus, in this method, a new plant is produced. Then comes the third method of artificial vegetative propagation, that is grafting. Grafting is generally practiced in plants that cannot be rooted easily or which possess weak root stems. Example of the plants which exhibit such a method are mango, apple, pear, citrus, guava, and many others. In this method, there are mainly two regions which help in carrying out this method. First, stalk, and second, scion. Stalk is the main supporting plant with the help of which grafting is carried out. That is the main supporting plant. And scion is the plant which has to be grafted or which is being grafted. Generally, a plant possessing higher and desirable characters is selected as the scion. In this method, generally a union is established between stalk and scion of two different plants. Now these plants can be either same or different kind. Such a union is established between different tissues of the different plants. This process can be induced more successfully among the plants which possess meristematic tissue. However, plants with permanent tissue can also be induced with the help of grafting, but the one that possess meristematic tissue are more preferable. Therefore, plants with meristematic tissue can be obtained easily with grafting. This figure depicts the grafting method in plants. As it can be seen, the stalk or the main supporting plant from which scion or a part of the tissue is extracted and inserted into the other plant, with the help of which new plant is developed and after studying the grafting method, there are various types of grafting that occurs. For example, bud grafting, cleft grafting, these are the main methods. Apart from this, there also occurs other methods like side grafting, Wedge grafting, tongue grafting, and crown grafting. However, out of all these, bud grafting and clift grafting are the most important of all, which is mostly carried out. Now, after studying the methods of vegetative propagation, whether natural or artificial, we'll study its importance or significance, that is, its advantages. By this method, improved varieties of plants can be obtained easily. This method is rapid, cheaper and easier compared to seed production. By this method, the common infections that are present in the plant can be easily removed. That is, the common infections that are present in the mother plant. The plants which have reduced power of sexual reproduction can be reproduced by this method very easily. Therefore, plants with reduced power of sexual reproduction can be reproduced with this method very easily. With the help of this method, the same characteristic of the plant can be obtained as that of the mother plant. Therefore, plants with similar characteristics and in large number can be obtained easily. Moreover, the plants with long dormancy period or seed viability can be also easily obtained with the help of this method.